Hello.
Hi, it's Elizabeth.
Hi, Elizabeth.
Hi. I'm joining this week.
Thank you. So I have a quick question. The
Sure.
supplement, the document that you sent me,
Uh-huh.
is it okay if I share it with the other CDT members?
Um, I think so. I cut all the financial stuff out because I wasn't sure if Lucilla wanted me to share that part. So I think it's probably okay.
All right. I was going to send it out, and then I was like, oh, you know what? Let me check. So I would have sent you an email later on in the day. But
Yeah.
anyway, since I had you, I thought, okay, let me ask.
Yeah.
Hello.
Hello, this is Nansu from UCSD.
Hi, Nancy.
Hello?
Hello.
Hello, Anu.
Hi, Ron.
Hello, Anu. Hi, Ron. This is Phil from Oxford.
Hi, Philip.
Hello.
We just wait for a couple more minutes for some more people to join us. I think Jeff is going to be a little late today. He's at some workshop at NIH. That's why I didn't see you. I know I thought that Sean.
I should cook from my office. I'm doing some stuff on my computer.
Um, maybe you could get started. I still hear people calling in. Hello.
Hi, this is Wei Wei.
Hi, baby.
Hey, Anu.
So Jeff said he will be a little late. He's at NIH. He's attending a fair data workshop.
File? Oh,
Fair. And
we asked about the thing tonight. Do you actually want to go? For what? To go to reception. So 4 o'clock there's John Glasgow.
I didn't get anything.
Over there. I don't think they actually send out the email. Right? Why John is saying encouraging you people to go? I don't know.
So Shaoling and Drilling are a little busy, and they won't be able to come in today for the meeting.
They do more.
They're working on the challenge stuff.
So finished.
They want them.
They don't come to then.
I think today
They can wait for 1 hour. I think.
it's okay.
Anywhere they.
Okay.
Hey, this is Jeff.
Yeah. All right, Jeff is here.
Oh, yes, okay.
Yeah, I'm on break, so I only have like 20 minutes probably.
Okay. So then we'll probably talk about. Let me. Right now we have in the room Shaoling, Willing, Hua, Trevor, Elgin, and Nina, and on the call we have Sri, Elizabeth, Huni, Ian, Nansu, Philippe, Ron, Wei, and Jazz. Did I miss anybody?
Hello.
Okay. Hi, Alejandra.
Hello, how are you?
Let's just get started. First, we'll have updates on action items, and then this is from last week's, the data type ontology that we wanted to talk about, and we also wanted to briefly discuss some of the issues which we thought we need to bring it, that were reported in the prototype issues that we wanted to bring before the CDT, before any resolution is taken on those issues. And we also want to talk about inclusion of more repositories into DataMed, the plan and course of action, and if time permits, then updates from all team members with respect to what they're doing. So the video, Jeff?
Yep. So again, the student's been working on that. So let's see. I'm trying to figure out when, because next week I don't think I'll be on the call because I'm at NIH again. So perhaps we can sit down, have them come on the call. So in 2 weeks.
In 2 weeks?
Yeah.
For the link out discussion, Ian actually was able to get in touch with somebody from who handles link out, and he sent me the contact information today. So Shaoling and I will get in touch with that person, and we will start looking into how to have DataMate get into the link out from PubMed. The other action item was to send out the survey to mailing list of prior users of DataMed. I did that last week. I think I did it. I sent it out to all the DataMed version 5 release mailing list that we had, plus the current users. and that was about 400 and odd people. So we hope we'll get some response from them. Another action item was to generate a help and a FAQ page, which we haven't started on, which we will start up on soon. One more action item that came out of the meeting last time was to have a robust server host BioCaddy at UCSD. So I think Jeff, you were supposed to talk.
Yeah, I sent an email, but I've been traveling, so I haven't been able to loop back around. So I'll do that when I get back to San Diego.
All right. So for the data type ontology, we have had a couple of issues reported where they've been asking about what ontologies we've been using to designate data types in DataMed. And so we haven't actually had, we haven't really used any formal ontology as yet, and this definitely needs to be worked on. So we had offline, or I think Philippe had also mentioned this earlier, that it would be useful to have an ontology or generate an ontology for data types. And we also had an offline conversation with Alejandra where she said that they've initiated something. So she shared that. Okay, sorry, give me just a minute. Let me pull that up. So there's a spreadsheet. I think I have too many spreadsheets open.
Yeah, so the spreadsheet I shared with you, it's not just about data types, but other vocabularies that are needed for other qualifiers that we've been discussing. But about this task, it's something that we are working on, and it will be one of our tasks on the remaining on the project.
Okay. So
yeah, and I know that for some of these, for some others as well, for some of the privacy, I think there was an additional entry, additional. So we'll just have to make sure to double back and check all the current sources that have been mapped to the latest DATs to make sure that we have all the entries entered on this spreadsheet as well.
Yeah.
So you just sent this out, or?
This is linked at the moment from the DATS spreadsheet, but yeah, we can share this. At the moment, as you see, it's just a list of values.
Right.
But we can complete and then find definitions and ontology terms where available. So. Yeah,
so because as people are going through the data sets and mapping to 2.1, they should add, if there's something that doesn't fit, they should add it in here and somehow note that somehow.
Yeah, sure. So yeah,
yeah,
we can distribute this spreadsheet. No problem.
Yes, and also this is Philippe, and there was a spreadsheet circulated by Anu last week. I think it's linked through the slide to a Google Doc. But I guess the idea would be not to necessarily make a new ontology, but create an application ontology that would consume existing terms either for EDAM, BRO, the one that are used by NIF, and see what is the most relevant element. I guess the collaboration would also be possibly with the search engine if we need to do mapping and expand a synonym list for different resources.
So some of the concern that we've seen being reported is for some of the repositories that have mixed data types, we've said that the data type is unspecified, and the comment from the links folks, I think, was that they would rather, since links had each individual data type being tagged with the data type, they would like to see that information being reflected in the data types that when we display links data in BioCaddy, in DataMed.
Yeah, so that we can look at how the link state is being treated, but they mentioned that it's in the metadata.
Right. So Kathy, who put in the comment, actually did mention that it is available as part of the metadata. I can pull up the specific issue.
Yeah, it's a Git issue 143, I think, right? Yeah.
Yeah.
Okay.
So they call it the subject domain area, and they have the different data types put up for each data set.
Okay. Yeah. No, we can take care of that then.
Yes, because every repository, I think that there is also work that has been done under biosharing, for instance, where Pete Michaelton has been remapping or annotating consistently across the different repos. So again, taking advantage of all this work and to provide the kind of mapping between the tags used by the native repository, the primary repositories, and the annotation that we have, we could definitely come up with a kind of network implementation that would deliver a good coverage.
Right. So for example, here in the example, they show transcriptomics, but they match it to gene expression, which I don't know if that's what we want to do. If we want to keep it rather at transcriptomics, because gene expression is fairly broad.
Transcriptomics is pretty broad too.
Yep. Yeah.
Does data type follow through to, with gene expression, you perhaps want to know what platform, or maybe that's too specific. It's done on, because platform with gene expression could be right down to what chip is used. But, at some level, you want to know whether, are we talking about microarray data, or are we talking about some other kind of data? But it's a question of how specific within that one wants to get to what the kind of...
So Ian, can you speak up a little bit?
Sorry, I've got some allergies going on.
Oh, okay.
Yeah, there's a question here, and data type in some ways ties in with platform. Both transcriptomics and gene expression are very general level, but what is probably, I'm going to speculate, is a more useful thing is to say, well, was this a microarray of some kind? But then, is it pushing it too far? Some people push that to say, well, what's the chip that was used in this study? Or what kind of assay technology, assay platform was being used? I put that forward as maybe being the kind of detail that one would want to get to with what we're calling data type.
This was certainly among the use cases that were initially specified. And I guess we will have to decide what is the priority here. But in a way, I think when we design the model, the DATs, we cater for that level of detail.
I think there was also an issue where they were talking about how Bioproject also specifies data types for each of its data sets, and we are not mapping that right now. But then I guess that's something that's going to get mapped when we have everything mapped to that's 2.1 in version. When we were doing version 1.0, I think we didn't map it, or maybe it was 0.5 that we didn't do the complete mapping.
Yes, that's a very good point, and also because we've recently been in touch with the European Nucleotide Archive dealing with submissions directly. And basically, they used to have under the study object, which is now replaced by the project object, kind of a study design type with a set of control vocabulary. But they are re-engineering the list so that it is a bit more consistent internally. And so we will have to follow up with the group of bioproject to see the kind of terminology they intend to use and specify.
So, Felipe and Alejandra, then you would be taking care of generating or bringing in a set of ontologies for the different, I guess, values that we want to have in DATs, right?
Yeah, I think, what we intend to do is to first specify the approach as we did with that. So one would be to revise which ontologies are relevant, and as it was being discussed now, to discuss as well what different repositories are using for annotating these kind of things, and then try to come to a consensus on what is the best terms to use. But yeah, this is what we will be doing as the next task.
Okay.
And I would add that it will be necessary to closely liaise with the different groups and under DataMed, so with Jeff and Hua, just to make sure that we make the most of understanding which databases are and pipelines are in priority in the process, and we have a good overlap and good coverage for all these elements and good coordination.
I mean, I'm just quickly looking at bioproject, and what it's got is probably insufficient. It's got a list of 26 data types, and they're really focused around genomics and omics technologies. So, and we need something broader than that.
Absolutely, yeah. And sometimes they seem to be a bit ambiguous in terms of cutting across either by platform or application purpose. So we run into issues in categorizing based on the typology.
Okay.
Yeah, and actually yes.
Sorry, go
exactly.
ahead.
What I was going to say is that this list that you share with us, that and anything else that the curators find while doing the ingestions will be really useful as a way to evaluate and make sure that we cover all the data types that are required.
Okay. So maybe we could get back to this in another 2 weeks' time or in a month's time.
Yeah, can the link to that spreadsheet be sent out so that we can then forward it to the people doing the curation?
To this one or
no, the other one, the other sheet, yeah.
Yeah, the other sheet. Sure, Alejandra, do you want to maybe put that spreadsheet in under BioCADI CDT folder, maybe?
Yeah, sure.
Thanks.
Or is that the same where the DATS documents are located at the moment, or?
So there is BioCADI has, under BioCADI Drive, there are like different folders for the different. So for the most part, I've been storing, list of repositories and everything else in a folder which is called resources. But since this would be something that's directly relevant to the CDT on which different people are going to be working in, I thought maybe we could just put it under.
Okay, yeah, sure. I'll put it there.
So Anu, can we go to the data set processing?
Yes.
I have about
the list?
Yeah, well, because I have 5 minutes. They've actually started their working group, so I have to jump back in.
Okay, so this one?
Yep.
Okay.
So I know from the prior call, I know that UT Health and UCSD, the DBMI, so Hyunyi and Ansu, that you were going to go through some of your older ones to update them and get used to the DATS 2.1. And the key thing is making sure that for the new repositories that we're bringing in, that we have the connection of how to get the metadata, because that has to be hooked up so that we can actually get the system to ingest the metadata and get it ready for the mappings. So once a site picks up a new repository to do, the key thing is the first thing to do is register. With either we need to create a ticket of where we're going to track these repositories as they're being ingested, but we need to sort of get that information on how to extract the metadata from that repository. Is it a file download? Is it a web service? Is it OAI, PMH? What is the way that we're getting metadata from them?
Right. And is there some place here that we can put that into or no? Oh, I'm sorry.
We could either that or we do tickets in the where we're managing data sets anyway with the data set requests. We can just add new sources in that private repo. So that we have a place where we can store the conversation. And then this spreadsheet is when you're doing the mappings, that we document what elements are being used and also what elements are. So if you scroll down a little bit, you'll see a purple one on the left, right there, primary, or formula. So formula was in PDB, and it was something that the curator deemed as sort of an extended attribute. And so this is sort of, with DATS 2.1, we have the core and the extended values, but then there's also the ability to add sort of a repository-specific attribute extension. And so we're noting that in this sheet so that we can keep track and then also give feedback to Alejandra and Philippe, sort of what the repositories are providing.
So, Jeff, can I ask a quick question?
Yep.
Would it be possible to somewhere have a legend or something to show what the different colors mean?
Oh, sure, yeah, we can do that.
Yeah, because for some of the people who are doing the mapping at our end, they don't necessarily attend this meeting.
Yes, we can add that, probably right in that line 2, 3, 4 in the beginning.
Yeah.
Yeah.
Thank you.
Jeff, I have a quick question. So
yeah.
which GitHub repository should we use? The data pipeline or the submit repository management repo?
Potentially the repository management one, because that's the one where we have all the repositories listed that we're going through. So that might be a good way to, because then everything is in the same repo and it's easier to keep track.
But this is only for those that were submitted, right? Not for the ones that we are ourselves going and bringing in the information.
Well, yeah, I mean, it's ones that we're working on. So the question is, the other ones, do we add them to that same queue? Because it's the same work list, right? That's the total work list of what everyone's working on.
And many of the repositories, actually most of the repositories that we have been working on, they do not provide API or download capability of the metadata. Basically, we just scroll
the prior ones, right?
Right,
but
right.
I think for the new list, we're going to try and prioritize ones that provide metadata.
Right.
So should we just say ready for review? So what does this ready for review mean?
So the ready for review is when it's the index is published to the development server for review. So we can, that's basically once the transformation has been submitted and we run it through the full pipeline and then index it, and then it can be reviewed, and then we can release it.
Okay. So just to clarify, Jeff, then if we are doing transformations, which were what we did previously, right? Like for example, we are working on Dryad, and I think Dryad it has been completed.
Okay. Then just check it in, and then again, we should probably create it
in the data pipeline one, right?
Yeah. And then we just have to figure out where we want to coalesce the tickets. We could do it in the data pipeline one as well. I was just thinking since the sources we're doing are already in the other repo, for the high level source information, that we keep that all together, since then there's one repo for people to go to know what sources they're working on. Because as we get more sources, we can add them into that repo so we can submit them, right? So in a sense, we submit them to that pipeline, and so the tickets will sit there until they're picked up, by a group that's ready to work on something.
Okay.
And then the other repo is sort of for the transformation files and all that. Any edits or changes or issues can be raised in that repo.
Okay.
So there's one for management and one for actual issues.
Got it. So for talking about this, I guess some time ago we had talked about discussing how many repos we want to map per week, and having everybody kind of report on where they were at that point of time, and what their plans were for the next week, and what they were able to accomplish the prior week. Maybe we could have like 5 minutes set apart in the CDT to discuss this every week.
Yep. Do we have a list? I mean, I know that you had someone looking at the repositories to look which ones provided metadata. Do we have that list yet, or because that would be sort of the starting point, right?
So my thought was even those where we are doing the mapping for the prior repositories, like from 1 to 2.1, right?
Yes, yeah.
Even those could get included, and those are first priority. And then we have the remaining list which came out of Commons, and we have a list that came out of our previous plans. Like, I always forget to share the screen. So this is the primary list that we want to go through and get mapped to that 2.1 and get it ingested. And once we are kind of more or less, and this also includes the list of repositories. So Sanda has been entering, or rather I did this all, but I've asked Sanda to actually enter in the repositories as they're submitted in our repository submission form here, so that we do know which team is working on it and whether it's been mapped or not. So this will be our primary list to work with. And then we move to the other, the larger list, which has about 120 or so repositories.
So
Okay,
also,
and was there someone who was? I thought there was someone looking at the metadata status of the repositories. Was that in the other sheet or?
yeah.
So what do you mean by? Oh, this information?
Yeah. Okay.
Yeah. We have somebody looking in the other. So he's filling it up in the other spreadsheet. But for the relevant repositories, we are bringing it over here anyway. So we have all the information in one place instead of having to go to multiple spots.
Okay.
And if this was submitted via GitHub, then the issue is also linked. You can see.
Okay, because then we should figure out which ones of the ones that are upcoming that have that have the metadata available.
Okay.
Because that would prioritize which ones we're gonna pull
immediately, right?
Yeah.
And you also, for the most part, have the teams assigned already here. So,
yep.
It gets easier then to do it.
Okay.
And this is in the BioCaddy folder under the resources folder. It's called repositories to be indexed for DataMed version 2.
Okay.
All right. So Jeff, was there anything else that you wanted to talk about?
The main thing is for the ones that the mappings for 2.1 is also for any of the sources, just to confirm with us the connection information for the metadata, so that we have the latest information for how we are grabbing the metadata, just to make sure we have the correct version of that.
That would be the information that's outside of what's here, or is this what you're talking about?
Well, if it's a web service, it's the web service URL, the latest one that you're using, and the parameter, just to make sure that the ingestion method that we have set is correct.
Okay.
And that's why potentially opening up a ticket for them in GitHub is easier to convey that information then, because then we have a record of it.
So this is, I guess these are all the earlier ones that we use, so you would want this to be double-checked again?
Yeah, so because for some of them, and some of the new ones, like I know that, for example, the life science database is done. That's sort of almost ready to go, just to make sure that we have the correct information, for them. So.
Okay. Okay, so for each of them, then we will open a ticket in GitHub.
Is it okay? Sure? Yeah.
So what do you think is a reasonable number of repositories for each site to map per week?
Given that the metadata are available, right?
Yeah.
So in the last meeting, Jeff kind of, correct me if I'm wrong, Jeff kind of mentioned that one repository would take about 3 days.
Yeah, that was sort of
kind of our estimation too. Yeah. So 2 repositories per week.
2 repositories sounds okay.
Yeah, but again, that's if the metadata is available, because otherwise we have to write crawling scripts.
Oh yeah,
and things like that.
Yeah.
Yeah, and that takes a long time.
Yeah, 2-3 weeks.
So.
2 weeks, yeah, at least.
You haven't started with any new repositories.
I got to run, guys. Okay.
So we are wrapping up IRD and Viper, although Viper might take a longer. And we started working on bioconductor.
Okay. So we also haven't run the complete cycle for ingestion yet. We're still really also still learning. Let's try to learn the whole process as soon as possible so we can really start working on it.
Yeah.
Okay, the two repositories.
Sorry, did I hear right that bioconductor is being considered?
That's what assigned to us, yeah, UCSD DBMI.
But the content is not necessarily data sets.
It's a software, yep, that's what we found. It's a collection of software.
I'm glad you asked that question, Alejandra. That was occurring to me as well. So I was trying to use my imagination about this, and I do remember that bioconductor is based on R, and one of the things that when you download R or when you download bioconductor, there are data sets included, and many of them are there for the purpose of demonstrations or testing things and so on. But people also can distribute a data set as an R package. And one of the interesting things about it, and I got into an email exchange, and it's probably a couple of years ago now, with Martin, I'm blanking on his last name, that's at the University of Washington. There is a scheme within R and bioconductor for describing a data set, describing the metadata about a data set, and I thought that was interesting and potentially a way of self-documenting a data set. So there may be something here in terms of data sets, but my initial reaction was like Alejandra's, that bioconductor is mainly distributing software as opposed to data sets, but it's not entirely devoid of data sets.
I
exactly.
think
So there are data sets included, but most of the main purpose is to distribute R packages for analysis and annotation and other functionality, but it's mainly software.
on the other hand, for DataMed to demonstrate that it can handle software as a digital object for indexing is probably a good thing in the long term.
Yeah, but it's a bit of a leap, Ron. I mean, sure, in the long term.
A little bit.
We haven't got the metadata. There's a lot of work that needs to be done before we get onto that. It's not just simply a case of, indexing.
No, I agree. I know. I'm somewhat familiar with R and bioconductor. Yeah, I agree.
Okay, then should we just drop it without spending more time?
Bioconductor was one of the repositories that were submitted, right?
Yes, and passed our review. Apparently, that's why it gets assigned to the team.
So the EC actually signed off on it?
Who was it submitted by? Do you remember, Anu? I mean, just don't want this to come out wrong, but it might be worth getting back to them and saying, what were you thinking? Except tone of voice and the way you asked that question has to be done carefully. No, asking someone, well, you suggested BioCaddy, what was their vision for that? How did they see that would work, and make sure we're not missing something here?
I don't think the EC looked at this as far as I can make out. I'm just looking at the So we've just said that it will be reviewed by our team and we'll be in contact with you to inform you of our progress.
So I see Sean's no, is this Sean Davis?
Let me see. Yes, it is Sean Davis.
Sean has been asking me about API access to BioCAD. He's here at NCI. I can ask him about this.
I guess it could be anyway an interesting use case to test the DATs, because Bioconductor is a collection of software packages, so the data type would be that. And as a use case, I guess it relates to supporting, once we have identified or discovered a number of data sets, can DataMed provide or suggest a set of tools which may be used to analyze those data sets. So that's long-fetched, but that's possibly a legitimate demonstration of the usefulness of DataMed.
So I guess that's what, so we had a meeting, Philip, I don't know if you were on the call or not, that was yesterday, where there's a supplement that is getting funded that will be between Links and BioCAD, where one of the aims is to have a plugin that could potentially have links to data analysis tools, etc.
Mm-hmm. Well, I guess we could test with a couple of packages and see what we can pull. I mean, not go the full Monty, but see a kind of range finding exercise, how far we can stretch the DAX to support all this.
My view is that, I think there is in the pipeline many other databases, right? Including, for example, we had discussed in the past scientific data, so including journal data set information. In a way, I do think that in the future it could be included, and actually what we have on that can cover part of that as well. But I don't know, my feeling is that it would be better to prioritize what is data.
Yeah, I agree.
So on the same lines, we have another repository that's submitted to get ingested or included into DataMed. This is SimTK, and Xiaoling looked into that a couple of days ago, and the issue there is that, again, SimTK has projects which have simulation models, source code, etc., and not everything necessarily is a data set. So one of the questions we had was whether to include everything or just those projects that have a data set associated with them.
I would say just the data set, but the way this conversation is going, and one of the hypotheses around, or certainly one that occurs to me with regard to indexing software is, there's a certain amount of common metadata, whether you're indexing software or data. Both of them have an author, right? Both of them have a date of creation or some such date, etc. And at some point, you ask the question, what's the difference between the metadata that you need for data and the metadata that you need for software? And if they're not that much different, then maybe with the DATS model, we've got something that would be very close in terms of indexing software. And perhaps having some awareness of that would be a good thing to have.
Okay, then maybe we've spent about 2 or 3 days already, so maybe we will spend a few more days and just complete this case. How about that?
But looking at what you've written here, you said the basic unit is a project. Well, what's the metadata about a project? And if you were to index their projects, how would that fit into the metadata model, into the DATS model?
Right. We'll have to look into that. We can do that. We can ask Pratik to look into it. And if we have a lot of problems mapping, if we don't, then we bring it up again and we can decide what we want.
We haven't contact the data yet. So we ask Pratik to look for first.
No, you can actually contact the submitter.
Okay, so see if they have it.
If they
provide
want
the metadata.
Yeah.
Okay.
Okay. So we have about 10 more minutes left. I just quickly wanted to go through some of the issues that have been reported, for which we just want to make sure that we understand or what we think is okay or is what should be decided, or we probably should go over and decide this. So one of the comments that we received was that the prototype has no support for collaborative research. It's meant for an individual. There's no workspace for collaborative analysis. But this is out of scope from what I think. Is that right?
I mean, this is a question of interpreting what we're seeing here. And from what I see here, my interpretation of it is, yes, it's out of scope, but one would need to say, why is it out of scope? I mean, my reaction to this is, I mean, they're asking that it be a workspace in which to carry out analyses. Well,
right.
Yes, but that's the commons. That's not DataMed.
And also we don't have, well, it depends, we don't have actual data, right? So.
No, this is kind of a myth. It needs gently pointing out what DataMed is. Because
I think Jeff did.
It's way off. Taken at face value, the words here do not describe anything that BioCADI DataMed is supposed to do.
Yeah, but Jeff did reply back and say that we are not going to have data in DataMed, so it would be kind of out of scope. But I just wanted to make sure that we say that, and we could then potentially close the issue after getting in touch. I think we did get in touch with them already. So another issue which had been raised, which came in directly through the contact us form, was this issue of having a plugin for gene transcript, protein, or metabolite queries. So this, to some extent, I think will be accomplished via the supplement with links, if they do get the plugin that they were talking about yesterday implemented. So we could mention that.
So back on 63, the person who raised it is someone at UT Health.
No, so that was entered in by us because we got it through a direct contact us form. So there's no way for what you call DataMed feedback currently was just us manually doing it at that stage.
All right. So do we have any traceability back to the person who raised this?
Yeah, we do. We'll have to go back and look. We probably have a spreadsheet where everything is stored, or a database where everything is stored. I think this plugin question came from Andrew Su.
See, the question with 63 is it's so way off target for what BioCaddy is. When I asked the question, how this person, if they had an awareness of what the whole BD2K program is and what the commons is, they would probably be able to see that in some way we're trying to address what they're talking about. But it would be good to know who it is and why that information, so we can work out why that information is not, that message is not getting through to people.
Okay.
So if you can let us know who raised 63, that would be good. Thanks.
All right. So so these were all, I mean, since they're really early numbers, you can see these are all issues that are pretty much raised, I think after we released version 0.5 probably. However, this is again, I think, asked by Andrew Su, and the question was whether we should allow people to search on any column by clicking the column header. I remember vaguely that we discussed this once and said that it's not a priority. Or is this a functionality that we want to implement?
They're talking about sorting, right? Not searching.
No, it's
they're
searching.
talking about searching, not sorting.
Not sorting.
I don't think
because this is really weird.
Searching those fields is already searching.
The common functionality, if you've got a table of data, is to be able to sort by clicking on the column header.
No, but they're talking about searching, right? It's not sorting.
No, I see that. It's just weird.
I think there are a couple of websites that I've seen that let you sort on columns like that.
Yeah.
I mean, not sort, search on columns. I've seen those. They're not that many. I think I've seen probably one or two.
I think it's okay right now.
It's not a priority for us. But do we want to implement sorting? But in order to be able to do that, the method not...
It will be difficult to sort. Some of them are description. How do we sort it?
Right.
Based on what, right? But if anything can sortable, if it's a time, we already have a sort time.
We have implemented, I think, sorting by date, title, and authors, right? Citations.
Yeah, so I
So
think
these
that's
are the
pretty
four
much
fields
a sortable field, like the authors.
So I think we just say that it's not a current priority. We will look into it later. So the issue 45 is something that needs to be, I guess, resolved. Eventually, this was brought up by Greg Farber, and it was a question of whether he used quotes for searching or not, and we didn't hear back anything. I guess, Ian, you did try to contact him a couple of times, right?
Yeah, this is really old.
Yeah, it's very old.
This thread has gone dead, hasn't it?
More or less. I'm just trying to figure out if it's still something that we want to, resolve and close, or
you're putting me. I'd have to go. There's a dozen things here. Let me look at this one and give you my take on it. I mean, I weighed in on the thread before, so.
Okay.
It'll take me too long to answer the question you asked. You just asked me.
That's about it actually, in terms of what we wanted to talk about for today.
Well, that's good timing.
We finished 2 minutes early.
Well, Anu, let me take up about a minute and a half.
Okay.
One of the things that Ian and I and some of our colleagues have been talking about is how to get a handle on practicing scientists and how they're interfacing with DataMed and the degree and extent to which it is useful to them. And so we'd like some thought by the core development team about how that might be done.
And you have 1 minute.
Well, no, obviously there's not enough time
kidding,
now.
Ron, I'm kidding.
Yeah, I know. But it is something that we'd like to have you be thinking about, turning around your minds and see, because the people who have been chosen thus far are the people who have actually stepped forward. Earlier I heard a figure of about 400 who have been contacted again. But the community is broader than that, and so we'd like to see if there's some way that we can assess that broader community of potential users and their experiences with DataMed.
So the question is about how we engage more users, or?
Well, engage more users, but in a way that has them tell us how DataMed is working in their daily lives.
I mean, I think this is sort of bringing up again, I think, something we talked about briefly last week. But it's, what it's highlighting is this issue is still there and it's still of concern to us. And I think, last week's discussion, I think we were still, looking for ideas on this, but I think, Ron's emphasizing again that this is important.
So basically the user study maybe will tell us some information, but it sounds like we want something more than that. Basically we want to hear some kind of success stories of using DataMed, how exactly can help their research, right?
Yeah, I think, that first story that shows up wherever that says, I went onto DataMed, I found this and this. It helped me with my research, and here it's being published.
Okay. So finding the data is really just the first step, but I mean, before they really go ahead and
Sure.
use the paper.
Yeah, no, I understand it. It's not going to happen immediately.
Well, but I think what can happen immediately, and looking at some of the things that we see in GitHub, I mean, what's refreshing to me about that is we're seeing somebody there actually trying the application and giving us their feedback. And at some level, we don't want much more than that, as long as what they know is this thing's supposed to help them find data. Does it help them do that or not, and what are the issues involved in it? And we can overanalyze this, but I think just engaging a lot of people in doing that will give us useful feedback.
Yeah, I still think one way to assess the success of BioKD could be really if people think about searching data sets, well, they first think about BioKD, the DataMed as a start point. So I think it's actually important for us to expand to like include major data repository, then we can really promote this tool and link it to a lot of other like PubMed or any other things. So then people starting to have this habit to when they think about searching data, they go to DataMed.
All right, I have to sign off.
Yeah, just food for thought, and maybe not next week, but sometime in the future we need to have a detailed discussion.
Okay.
Okay, thanks. I took more than 2 minutes.
Okay, thanks.
All right.
Thanks,
Bye.
everybody.
See you next
Bye.
week. Bye.
Thank you. Bye.